So I will be talking on a very, very important, and whenever your title is advances, I'm talking about the advances, recent trends. Because we know up to the time about the different types of waste created by the human being. And we know that a human being always wants development. We are after development. And as a result of development, the waste is created. After independence, 1947, we have made various laws and rules regarding treating different types of waste which are causing nuisance. The first law was regarding the water pollution, sewage, then municipal waste, solid waste, hazardous waste, metallic waste, many other industrial waste. There were rules. Then slowly other waste came into existence. One of them was a biomedical waste. And later on, very late in this century, electronic waste emerged in the earth, on the earth. And now people are worried because our central government in 2011 made rules regarding disposal of electronic waste. And how to treat electronic waste, that is a challenge not only in the world, also in India. And that's why I'm trying to focus the electronic waste. So in my first slide, I have told that when the world is trying to solve the problems of agriculture waste, municipal waste, plastic waste, biomedical waste, a new type of waste is rapidly growing now. And this waste is called as a e-waste. What is the e-waste? E-waste is also called as a electronic waste. And in short, it is abbreviated as a WEEE. Meaning is that waste electric and electronic equipments. The electronic goods which are not fit for their originally intended use is called as a e-waste. In short, discarded electronic and electrical equipments at home. And I will give the list of many electrical and electronic equipments regularly we are using at home. Now everyone is connected to my lecture either via mobile or via computers. What is it? It is a electronic equipment. Electronic equipments are so common now. Every person, every woman is handling electric and electronic equipment. At every house, there is a refrigerator, air conditioner, cellular phones, personal stereos, and a number of electrical, including hot air oven, mixer, washing machine, and whatnot. Many equipments which are regularly used at home and regularly used by you and me. Even in the colleges, in the offices, we are using computers, not only computers, we are using the hard disk. We are using different electrical and electronic equipments. All these are creating waste. Now, I'm not going in detail because large amount of waste is generated throughout the world. These are the old figures. New figures are again still astonishing figures are there. More figures. So the electronic waste is developed in many Eastern countries. What they are doing? For example, US, then UK, France, Germany, whenever their computer become of old version, they are donating to the Indian schools. And Indian schools are accepting these computers and these equipments which are outdated for the world, but which are new for India. And it has become a dumping land. India has become a dumping land. Important things in increasing the e-waste is low rate of recycling. And where I'm concentrating microbiological solution, how microorganisms can help in solubilizing the metals from the electronic waste. Again, I'm reiterating the very interesting and important point regarding the biotechnological technologies, biotechnological methods. It is creating a wealth from the West. Meaning is that biotechnology solution always create wealth from the West, and how wealth is created even from the electronic waste that we are discussing now. So the process of removing the components from e-waste is not economical. Then I'm going to the why electronic waste. For example, you are having pen drive. It is outdated. What you are doing? You are throwing on the garbage. For example, you are having a cellular phone. It is not out of use, but the old version. Whenever it is an old version, but because nowadays Android mobiles are there and everyone wants now Android mobiles. So what to do with the old mobiles? We are throwing on the garbage. And by throwing on the garbage, they are very hazardous. Meaning is that we are creating human health hazard by throwing these mobiles, these pen drives, these hard disks, or any other electric wire, including TV, including washing machine. They are responsible for creating human health hazards. Why? Because they contain metal, and these metals will be leached out in the garbage by different activities of microorganism. The metals leached enter in the environment. Then by rainwater, they enter into water bodies, ocean, river, lakes, well, and they enter into the crops. That is called as a bioaccumulation of metal. And from the crops and food grains, or from via fruits, via vegetable, they enter in your body and my body. And there is a bioaccumulation of these metals in our body. You will say that, sir, there is no bioaccumulation up to the time marked bioaccumulation. Yes, but this waste is increasing nowadays. And after 10 years, probably it will be the severe problem on the earth. So the toxic metal content is an important reason. Second is a low recycling rate. And they are causing human health problems. Not only human health problems, they cause environmental pollution also. Meaning is that environment, the animals, plants, microorganisms, all ecosystem, all biodiversity in the environment is getting polluted because of the electronic and electrical equipment. So they are dangerous. It has been proved. There are different ways how to treat it. We'll discuss. And one of the ways is a microbiological way. Now, for extracting metals from electronic waste, it is crudely handled. It is crudely dismantled. And it is some part of it in many parts of the world, it is incinerated. Incineration is not the proper way. It creates a again air pollution, toxic gases. But at the present, what we are doing with the mobile, with the computer, with the iPad, with other electronic and electrical equipments, we are dismantling it and we are incinerating plastic part adhered with these equipments. And most workers are child laborers. That is again the bad point in that. Although there is a rules and regulation in India regarding child labor, but in the villages, child laborers are working. They are not knowing, the children are not knowing what are the hazards of electrical and electronic equipment. You see, these are some pictures which I have given. Dismantling the computers and laptop and many other things. And these students, they are dismantling with the hammer. Hammer is shown here. And they are dismantling the all electrical and electronic equipment. It is a garbage of electrical and electronic equipment. What to do with the garbage? That is a problem in front of the government. You see again with the hammer, they are dismantling. Here also dismantling the wires and burning the e-waste to extract the metals from it. Although it is not the proper way, but it is a common practice. So let us come. How it is treated. I'm coming to the biological method, that is a microbiological method. But before that, already there are two methods which are existing. One is a physical method, as I have told you, manually or via machine. All electrical and electronic equipment are crushed and metal, we are trying to recover the metal from these electronic equipments. Second is the chemical methods. Even the acids can be used to dissolve the plastic and to extract the metal. But both these methods are costly and they are not eco-friendly. Now you see, in the biotechnology, very important word is a eco-friendly tool. Because human beings want to develop, but which development? Sustainable development. And in the sustainable development, eco-friendly is the major tool. So, whenever we want a eco-friendly tool, which is the eco-friendly tool? Biological method is a eco-friendly tool. In the biological method, we are discussing about the microbiological solution for dissolving the metal. Is there any organism which can remove metal from such complicated e-waste? And if yes, how? That is the question. And answer is that bio-leaching. Conversion of solid metal values into their water-soluble form. How? You see, bio-leaching is already established process. From the insoluble metal converted into soluble metal by oxidation. And after oxidation, metal gets solubilized and they can be separated easily. So, extraction of metals from solid matrix is the way. It is hydrometallurgical process where mobilization of metal, what is important? We want to mobilize the metal which is insoluble into soluble form. It takes place from solid phase to liquid phase through microbial activity. So, how it is possible? You see, we know in the history of microbiology, from the ores we can extract the metals. That is called as a bio-leaching. Many scientists have done the research on that bio-leaching process. Bio-leaching process is carried out by two different ways. What are the two different ways? Chemolithotrophic leaching and heterotrophic leaching. Biotechnology students knows very well what is a chemolithotrophic bacteria. These bacteria derive their energy by oxidation of metal. They doesn't require organic compound. Heterotrophic leaching Heterotrophic bacteria produce short chain fatty acids, and these short chain fatty acids are responsible to solubilize the metal, to mobilize the metals. So, two types of microorganism we have to concentrate. Chemolithotrophic, they are very difficult to isolate. Heterotrophic, simple to isolate. Here I have given some examples of chemolithotrophic bacteria, right from desulfurio, right from sulfolobus, leptospirillum, then the acidithiobacillus, peroxidin species, thioxidin species. There are different types of species which are responsible. They are chemolithotrophic. And the heterotrophic bacteria, right from Bacillus subtilis, Pseudomonas putida, Aspergillus oryzae, Aspergillus niger, and different species of Candida. These are heterotrophic microorganism and chemolithotrophic microorganism responsible for bioleaching. Let us concentrate first chemolithotrophic. In the chemolithotrophic bacteria, the carbon source is a CO2. Means they doesn't take organic carbon. They take carbon from the environment, from the air. And what is the energy source? Energy source is the oxidation of inorganic compound. They derive the energy. That's why they are called as a chemo. Litho means inorganic. Trophic means nourishment. Chemical inorganic nourishment. That is oxidation of inorganic compounds. Then Fe3, oxidized metal sulfide. I will explain this process in detail later on. Metals are dissolved in acid synthesized by organism. Chemolithotroph also produce inorganic acids. These inorganic acids are also responsible to dissolve the metal. The most important metal leaching microorganism use ferrous iron and oxidize it to ferric iron. You see. So this is a very important process. The ferrous iron is oxidized into the ferric iron and fix carbon dioxide. Many of these organisms produce sulfuric acid, as I have told you just now. Inorganic acid, sulfuric acid is produced by the microorganism. This inorganic acid is responsible for mobilization of the metals. Now, this is a very, very important figure. In this picture, I have shown the yellow part and a little dark part, blackish part. Yellow part is the environment and black part is the electronic waste. Now, the electron, we want to solubilize the metal from electronic waste. Now, the circular in shape, the cells I have shown circular in shape. These cells are microorganisms. They are bacteria. What they are doing? You see, very simple process again. The reduced iron is added in the environment. What they will do, microorganism, what they will do? They will oxidize ferrous sulfate to ferric sulfate. Ferrous sulfate is oxidized to ferric sulfate, and then this ferric sulfate is oxidizing the metals, reduced metals, insoluble metal, which are present in the electronic equipment. Which metal? Maybe copper, maybe lead, maybe silver, maybe gold, maybe any other metal which is hidden in the electronic equipment. Because in the electronic equipment, you might be knowing, there is a printed circuit board. In the printed circuit board, there are wiring of metals. And in that wiring, various metals are present. All these metals are solubilized by bacteria indirectly, not directly. How indirectly? First, bacteria will solubilize or bacteria will oxidize ferrous iron to ferric iron. Where it is carried out? In the environment, yellow part. In the yellow part, bacteria will solubilize ferrous iron to ferric iron. Then this ferric iron further dissolve the other metal, oxidize the other metals. For example, copper is there. The ferric iron will react with the reduced copper, will convert reduced copper, which is insoluble, will convert into soluble form, will bring into in leachate, and iron itself will get reduced. Do you understand? For the growth of microorganism, what is required? For the growth of microorganism, reduced iron is required. That is their energy source. Reduced iron, microorganism will convert into oxidized iron. Again, oxidized iron will convert into reduced iron, and reduced iron is again continuously supplied to the microorganism. Meaning is that it is a very, very interesting process. Only initially we have to add the ferrous iron. This ferrous iron is continuously generated. Then there is no need externally to add the ferrous iron. Ferrous iron is converted by bacteria into ferric iron. Ferric iron is again itself get reduced. Whenever it will oxidize, the copper, lead, magnesium, silver, gold, which are present in the electronic waste in the reduced form, they will be converted into oxidized form. And they will come into leachate. And how they will come into leachate? Here I have shown some examples. There is a chemolithotrophic bacteria. Some figures are there. And next we will go to the heterotrophic leaching. What is the process by heterotrophic organism? Heterotrophic organisms use organic compounds as a source of carbon and energy. What is the mechanism? Very simple. These heterotrophic bacteria they produce short chain fatty acids. Means heterotrophic bacteria produce organic acids, short chain fatty acids. Which acids? Acetic acid, propionic acid, malic acid, glutamic acid, isopropionic acid, and likewise many other short chain fatty acids. These short chain fatty acids will dissolve iron and they will convert insoluble iron into soluble iron. Meaning is that the leaching process or biotechnological solution to the e-waste is that chemolithotrophic organism and heterotrophic organism, both of them are responsible to solubilize the inorganic metals. Then heterotrophic bacteria, some figures I have shown. Here some fungi figures are shown. I will not go into here in detail. I will go directly to my research. But my research meaning is that one of my PhD student did the research. So I am going to the PhD student research. This is a process where bio-leaching was carried out in the mine, on the mine. So this is a mine. I will not go in detail with the mine. And which metals? It has been observed that cobalt, nickel, cadmium, antimony, zinc, lead, gallium, means like that, manganese, nerium, copper, and many other metals can be solubilized. And bio-leaching of metal is already established process, and from electronic waste also it is possible. One of my students did work on it. Why I'm putting this work in front of you? The reason is that much research is waiting for you. Young generation can concentrate on this topic, and very interesting because This is a burning problem in front of the world, in front of the India. So what work he did to develop the consortia of iron and sulfur oxidizer. My student tried to isolate chemolithotrophic bacteria and heterotrophic bacteria. First of all, he isolated metal tolerant culture, where from he brought the soil sample. He brought the soil sample from the mines, where there was a possibility of getting maximum organism. And he exposed electronic waste, and how he did the work, that I will tell you. And then he optimized the process. For that purpose, again, one more thing I have to tell you. I have given in the procedure, probably I will tell you. These are the printed circuit board. What he did, he crushed it into small powder. Even he used the entire printed circuit board also. But if it is converted into fine powder, it gives a better result. So the printed circuit board were crushed by mechanical means or simply by use of file. Then it was obtained, sieved. The powder was sieved, and after that, the fine powder was used for our experiment for biology. Then, before that, this is not visible, but I will tell you what is in the slide. Chemical analysis of electronic waste we have carried out. Why? To find out really the metals are present. There are many chemical analytical methods are there. So atomic absorption spectrophotometer is required. And by various methods, we analyzed it and we found that yes, in our waste, electronic waste, in the electronic powder, there are number of metals. Which metals are there? Copper, iron, zinc, lead, nickel, aluminum, silver, gold, tin, chromium, manganese, cadmium, silicon, and calcium. Many other metals were present, detected in the electronic waste. Then what we did? 15 gram per liter of scrap sample was added in 9K medium. What is a 9K medium? It is required, it is used only for isolation of chemolithotrophic bacteria. And plus 9K medium meaning is that, when the reduced iron is added in 9K liquid medium, the composition is given in my own book. I have written one book, Handbook of Media, Stains and Reagents in Microbiology. That is freely available on our website, www.microbiosociety.org. This book is being used by every microbiologist in India. So you can avail that book. In that, this 9K media composition is given. Then inoculum was added. Which inoculum? As I have told you, isolated from the mine soil and from the cow dung. Cow dung was also used. And from that, consortium was prepared, and 15 to 20% consortium was added in that. Then flasks were kept at 30 degrees centigrade for incubation on rotary shaker. Why for? In the powder, because there is a powder prepared from the electronic equipments. Again, I am telling you, you will ask me, sir, if it is simple to dismantle and to obtain these metals, why you are doing so tedious process? Again, I am coming to this. This is a eco-friendly process. And second, we can create a wealth. We can again obtain the gold from the west. We can recover the silver. Silver has an economical value. Gold has an economical value. Means what we are doing with the biotechnological process, we are preventing this metal to enter into the environment, and we are recovering this metal, and so that we can earn money, we can create a wealth by a biotechnological process. So here, unwashed, and I'm not going in detail. So Here we observed iron oxidizing bacteria in red color. They are increasing and the iron concentration is slowly decreasing. So the iron is oxidized and the reduced iron is converted into oxidized iron. And we observe after specific time solubilization of copper from print. We studied for copper first. Solubilization of copper from printed circuit board powder during 0 to 240 hours. We observed peak at 192 hours. Means maximum iron was entering into leachate. It was converted into soluble form from the non-soluble insoluble form. So we were successful in isolating copper. We were successful in isolating lead also. Then what is the process? Very simple mechanism. The mechanism is that iron Fe2, which is a reduced iron, is oxidized O2 plus hydrogen in presence of earth thiobacillus ferroxidans. Earth thiobacillus ferroxidans will oxidize Fe2 into Fe3. Later on, in the second equation, this Fe3 react with the copper and this insoluble copper is converted into soluble copper Cu2, oxidized copper, and iron will get again reduced. This process is an exothermic process. Delta G0 dash is a minus 82 kilocalorie. So kilojoules means it is a exothermic process. That is the beauty of again biotechnological method. These microorganisms are not asking salary to us. They are happy because they are getting energy source. And without any charge, without any cost, without any labor cost, they will convert inorganic iron into organic iron. So during the bioleaching process, a brown colored precipitate was formed and the content was determined by same EDAX method. Here, the EDAX analysis was carried out and concentration of different metals in our solution, in our leachate was detected. And the citric acid we found that is helpful during the process. And this is the conclusion that the bioleaching can be applied for recovering of metals from electronic waste. So bioleaching is the best process. Use of microbial consortia enhances leaching efficiency, meaning is that microorganism enhance the leaching efficiency as compared to pure culture. So we observe the consortia is better. Single culture organism is not better. Then as metals are having inhibitory action on the growth of microorganism, metal adapted culture should be used. So first of all, you have to isolate metal tolerant cultures. And higher metal extraction rates can be achieved by adding more percent of inoculum. If inoculum percentage is increased, we obtain better results. Greater concentration of Fe3 is increased, we obtain better results. So as soon as metal goes to precipitate, so early metal recovery should be done or some strategy need to develop to avoid or minimize precipitate formation. That's why we used citric acid, which will avoid the precipitation and cause more and more solubilization. The problem of precipitate formation could also be solved by the use of extreme acidophilic iron oxidizer. Let it be. Now let us go to the second. Here you see I have shown the 9K medium. In that the culture is added, slowly iron is oxidized and the color of the flask is getting changed. So my PhD student, he worked on that and he obtained the results after inoculation and after incubation. What are the colors? So the colors with a different concentration, 5%, 10%, 15%, and inoculum, and after incubation, what results, what color you are getting, that is shown in the flask. Now you see, iron isolation is a very, iron oxidizing bacteria isolation is a very difficult. It is not so simple to isolate the chemolithotrophic bacteria. And here, my student, he isolated chemolithotrophic bacteria. They are very pinpoint colonies. And we cannot subculture them. What we have to do? We have to take this agar portion and add again in the liquid medium. Then and then they will grow. It is not like other bacteria, like E. coli, like Bacillus subtilis. If we pick up them on the nichrome wire and subculture them on other media, they will not grow. They will not form the colony. So, iron oxidizers on solid media, he isolated the organism. These are the very fine colonies of iron oxidizer. In the medium, there is a insoluble, there is a soluble iron which is converted, insoluble iron is present in the medium, which is converted into soluble iron. So the color of the colony changed, and these colonies are visible on the solid medium. Again, enlarged colonies. I have enlarged the figure, the photograph of the colony. Here, enlarged colony. Here you see the enlarged picture of the colony. So we can get, and my student have tried to immobilize iron oxidizing bacteria. And because immobilization can cause a continuous process, it was a continuous, whether continuous leaching can be carried out. He has made an attempt. He has published a paper also, later work is. I'm not presenting his work today, that is regarding the immobilization. But what I want to tell all the students, this is a very challenging topic. Why it is challenging? You might have believed that every one of you, even the labor, even the layman, everyone is using computer, everyone is using laptop, everyone is using mobile phones. And regularly, new version of the mobile is coming in the market. And our young generation is attracted by the new version of the mobile. They are having new facilities. They are having updated version of the mobile. That's why old mobiles, although it is working, we are throwing on the garbage. That is a tendency. And this is, again, mind it well, central government has made the rules. You cannot throw your mobile on the garbage on the road. If you throw your mobile on the garbage on the road, you will be punished. If I take a photograph and I submit to the government, I can file a case against you, because every college, every university, every individual has to deposit the outdated computers, outdated mobile, outdated laptop to the collector, and the collector have a duty to collect all these electronic waste together and to treat or to dispose properly as per the rules. At present, there is a rule also, the burying all these solid waste, but burying is not the proper way. It is not the eco-friendly way. At present, these electronic equipments are buried, or these electronic, by Western countries, these electronic materials are thrown into ocean. In the ocean, they consider ocean is a dumping house. But in the ocean, the aquatic life comes in danger. Unnecessarily fishes, tortoise, and other aquatic life, the metal accumulation takes place in their body. That is not the way. Means it is wrong. to dispose, because in the Western countries, that is the way now. They are collecting all electronic equipment, and through the ships, they are taking in the center of the ocean and disposing all these electronic equipment in the ocean. So you see, students and all teachers, all researchers, I'm making appeal to you. We are a biotechnologist, and we should give promising solution. Only microbiologists and biotechnologists can give promising solution, sustainable solution to every problem of the world. I think 40 minutes were given to you, and exactly 39 minutes are over. I am very strict regarding my timing. Thank you very much. I'm very thankful to Nayyar sir again for giving me an opportunity, and Supriya ma'am, she was constantly in touch with me. Thank you all authorities for organizing such a beautiful event and arranging the discussion on various topics of biotechnology. Thank you. Thank you.